Thank you for inviting me to give one of the first in-person talks since the pandemic. And my name is Magness. I am a professor in the biomedical engineering department. I'm a member of the Center for GI Biology and Disease, and I am the core director for the Advanced Analytics course, which I'm going to talk to you about today. We haven't done one of these actually in 5 years. I was looking through my old slides and going over what services we provide. I haven't done that in 5 years, and we've added a lot more services since I gave this last talk. And so what I'm going to do today is I'm going to go basically just a high-level overview of the types of services we provide, how to use them, how to access them, the types of maybe caveat questions you might have about the services. And so I want this to be very informal. Please ask questions. If you want to, you can chat, and Amy Smither will read them out loud. I'll try to answer them. If you want to unmute and ask a question, you're welcome to do that as well. So a lot of people ask me, why do we call it the Advanced Analytics Core? And one of the reasons is because, like most cores, we offer technologies or services that are either too expensive or how to do it, the methodology is too hard for one individual lab to do. So we offer this through the core, but we do a lot of different disparate technologies. And so it fell into this idea of what is the most sophisticated kind of like technology we could provide. That was from genomics to protein analysis to single cell analysis to new organoid culture services, which we call culture services. So it kind of fell in this big broad umbrella of sophisticated technologies. So we call it the Advanced Analytics Core. So as I mentioned, we have a diverse range of services we provide. We provide FACS or cell isolation, fluorescence activated cell sorting, gene detection, either from generating bulk RNA sequencing libraries or single cell sequencing libraries through a number of different technologies, protein detection through kind of standard immunoassays or multiplex assays to detect proteins in solution, for instance, like cytokines. We also have a very cool technology to do single cell Western blots. So that's one thing that kind of goes in hand with single cell genomics is once you detect a gene in a single cell, how do you know the protein is expressed at that same level? And so we have the ability to do single cell Western blots. And then a very new service that we've just added in the last couple of years is what we call EP culture. It's basically a cell culture service that provides 3D organoid cultures and two-dimensional monolayer culture services using primary derived stem cell cultures, basically. And I'll go into the details of these in a minute, but that's just a very high-level overview. This is the AA core staff, very, very talented people. Carlton Anderson is Associate Director. He's a person that you will typically interact with at the consultation level for your particular project. Gabrielle Cannon is a genomics specialist. She's the individual who runs most of the single cell library preparations, the bulk sequencing, and also does the Milo single cell Western blot technologies. And then we have a brand new core specialist who is primarily in charge of the EP cultures, which I'll talk about in a minute. If you want to contact any of these people, I think the easiest way, and I don't want to put the whole website up there, but the easiest thing to do is just Google UNC-AA Core. And we'll take you to the web page, and you can see where all the services are. You can click on the individual service, you can see pricing, you can see what the technology is, who to contact for that particular service. So I'm going to start out looking to talk about gene detection. So we have basically 4 different ways we can detect genes or gene expression. One is through bulk transcriptomics. We have a microfluidic approach to generating RNA sequencing libraries. It's on a chip. It can do up to 48 libraries at any given time. And it's a very rapid, fast, and basically reproducible and robust system. So there's not, a human being is not doing the pipetting, a machine does it. So this allows you to really instead of doing multiple technical replicates, which really can control for human error, you don't have to do those. You can do biological replicates instead. And so this will decrease your cost quite a bit using this particular microfluidic approach to generating libraries. Remember, this is just the library step. It's not the actual sequencing. Single cell transcriptomics, we have primarily one platform we're using right now, which is the 10x Genomics Chromium platform. That's what most of you are familiar with. And we are exploring probably in the next week or two a new system from Parse Biosystems. The big difference between these two is the first one is a droplet-based technology, which you're all familiar with, that the chemistry is done in droplets. And this typically requires live cells. Most people use live cells for this, and that kind of limits a lot of people if you're off-site. If you have, a tumor that came yesterday and you can't actually get it to us live or fresh, it really limits some of the samples that we can process. The Parse Biosystems is not droplet based. Essentially, instead of doing the chemistry in a droplet, it does it inside of the cell. So the cell is actually the test tube. And that's the big difference. And that technology uses fixed tissue, only fixed tissue. So this opens up the single cell transcriptomics to a lot more projects than could otherwise handle. That requires live cells. Single cell multi-omics. This is a relatively new technology we've added to the core. It's again done through 10x Genomics, the company, and this allows you to do RNA-seq and ATAC-seq, or look at open chromatin regions in single cells at the same time. So this is a simultaneous multi-omics approach to look at open chromatin and associated gene expression patterns in single cells. And then the last thing is a little bit more conventional sort of gene detection method, which is qPCR. And so the more sophisticated part of this is we can do this in high throughput. So we can do up to 96 samples in 96 different genes at once. There's a bunch of different configurations of the number of targets you can detect or genes you can detect and the number of samples. And this is done through the fluid biomarker instrumentation. And I'll go over the details of this in a minute. Some people ask, well, if you're going to do sequencing, why would you do this? Or what if you're going to do qPCR, why would you maybe think about doing more, high-level single-cell approaches to detect gene expression? The big one's cost. How much does this cost? How many samples do you have? How many genes do you need to detect? There's a lot of caveats to how you approach the experimental design, and how you use that information to decide if you want to do qPCR or single cell RNA sequencing. And that's something that we discuss in depth in the first consultation when we come to the core. So I said before, we offer isolation, single cell isolation. One of the first steps for single cell transcriptomics is single cell isolation. That's one of the important things, to make sure that you have a single cell preparation. We have our own FACS instrument, our fluorescence activated cell sorter. It's the Sony SH800. If you want to look up the details of this, it has 4 lasers, and you can get about 8 color detection. There's a 2-way sort, so you can sort 2 samples simultaneously. It's core operated, so if you have samples that you want isolated, we can do that, or you can do it. So we have a training you can go through where you can learn how to use the instrument yourself. You can come after hours. If your samples are very precious and you're worried about us running them, you can run them. That does come with a cost for the training, and also you have to go through several iterations of samples to make sure that you're good enough running the instrument before we'll let you loose with the instrument. It's quite expensive. So we want to make sure you really know how to use it. It's also a BSL-2 instrument. So we have this approved to do human samples, to sort human samples, non-infectious human samples. I want to make that clear. So if you know that your sample has some sort of communicable disease, there's a number of them that you cannot sort on this instrument. We make you sign waivers that you understand that you know or don't know whether or not that sample has a communicable disease. All samples are treated, if you don't know, they're all treated as if they have a communicable disease. There's a special training involved in this too, if you would like to run BSL-2 or human samples. But this allows you to do a lot more things you could do outside of the core, because we have the instrument in a room that you can have access to after hours. If you get a tumor sample late in the day and you need to process it, it's something that you can actually train to learn how to use the instrument to isolate single cells for downstream analyses. So I'm going to now go into the RNA sequencing platforms that we operate. So this one is for bulk RNA sequencing, not single cell, but if you have a population of cells that you're interested in looking at gene expression patterns, this is where you can use this microfluidic approach to generate up to 48 libraries per chip. Some of the advantages to this, you can use very small amounts. It's a microfluidic approach. It's done on a chip. The volumes are very, very small. This decreases the cost because all of the chemistry you put in is in nanoliter amounts. So all that very expensive stuff you would use in a 96-well plate format is now shrunk down to nanoliters. So it decreases the cost a lot per library. It's about $65 per library. I already mentioned that because it's a microfluidic or automated approach that essentially a machine does, that there's no pipetting, really, where you can introduce mistakes between samples. And so this allows you to do more biological replicates rather than doing technical replicates to generate your libraries. Essentially, the workflow and how you would bring us samples and what we would give you is indicated by this schematic here. So you isolate your RNA. We do not isolate RNA for you. That is your job to do that. You can do the QC yourself, meaning that you can look at the RNA integrity number, look at the quantify it, things like that, before you give it to us, just to make sure that you have RNA, you have a sample that's going to perform properly. You drop that off to us, and we, we run the Advanta auto prep system. That's essentially the technology. It's called Advanta. It's from Fluidigm. We do a number of QC steps for you to make sure that before you go into the really expensive part of sequencing, that the libraries look good and that they're ready to be put onto the sequencing instrumentation. At this point, we can return the sample to you. So if you want to sequence using your own platform, or you want to send it away, or whatever you want to do, we can return the samples to you here and you can sequence it yourself. Alternatively, we can do this for you in the core, or we can send it to the high throughput sequencing core facility. And there are a number of reasons why you would maybe choose one over the other. One is how many samples you have, the read depth you need for that sample. Our instrument in the core has limitations on the number of reads that you can get per sample. So if you have many samples, that's something that we would outsource to the HTSF core, high throughput sequencing facility here at UNC. And the data processing, this is a big black box for most people. If you do not have a bioinformatician in your lab that knows how to process RNA sequencing data, that can be a problem. And so what we do is we've engaged with UNC Bioinformatics and Analytical Research Collaborative. And what this is it's a core of very smart bioinformaticians. And what they do is they know how to process this RNA sequencing data. And so during the consultation step for your project, if you do not have analytic expertise in your lab, we will hook you up with somebody through the BARC that will handle your data for you. And they work very close with us, the core, to understand what your experiment is, what your design is, what your goals are, all of those things. So that by the time the data come from our core and we pass it off to them, they've already had an idea, and you've then probably already worked with them to decide what are the deliverables that you want, what are the visualizations you want. And then once you work with the bioinformaticians from the BARC, you'll establish a relationship. And as you know, this is not a one-time thing. This is a thing that happens over months' time. And during manuscript preparation, the stuff you're generating for your just high-level analytics typically is not good enough to go into the papers and requires even more sort of data visualization, manipulations, and things like that. So that's all done by this BARC, and they do charge per hour fee for their services. But we handhold you through that process, and we get you to the point that you won't be scared of doing RNA sequencing. The next thing is the single cell part of it. So I was talking about bulk sequencing, the populations of cells. This is, we're now moving on to the single cell service workflow. I'm going to talk about the 10x because that's what we currently offer. The parse we are going to testing in a few weeks, and hopefully in a month or two, we'll know how it performs and whether or not we can offer this as a core service. Keep checking back with us if you're interested in the parse biosciences, single cell sequencing, the library preparation. Again, the workflow for this, similar to the bulk, but there's a few differences. One is we have a really extensive consultation. A lot of people want to do single cell sequencing, but they don't really know why. They think it's sexy, which it is. They don't really know how to think about how to design the experiment, because a lot of times this is very new for people. And there are a lot of differences between designing a bulk RNA sequencing project and a single cell RNA sequencing project. Even though you're getting gene detection in the end, there are big differences in how you think about how much you need to sequence, what you need to sequence, how many different samples you need to sequence. All of that is different for single cell versus bulk. So we like to have a very detailed consultation to go over all of those things that you want in the end to make sure you're going to get them. And we design the experiment right up front. It's much more expensive to do single cell sequencing than bulk sequencing. Just as an example, one sample of about 8 to 10,000 cells. I'm just going to give you some general ideas here because it really depends on your experiment. It costs about $75,000 to $8,000. That's from the start to the finish. You bring us cells, we give you sequencing data. Okay, that's not including the bioinformatics or anything like that. That's just this raw sequencing data that you give in the end. So it's quite expensive, right, for one sample. Now, what we've done in the core is we've piloted this. We got a grant from the CFAC here at UNC to basically develop a multiplexing approach that decreases that cost per sample by a lot. And essentially what this is just a cell hashing process where we take antibodies. We can take up to 6 different antibodies and apply it to 6 different samples. That allows us to pool those samples in the very beginning and apply those to this very expensive part of the technology. Then in the end, in the sequencing, after the sequencing comes back, we can deconvolute that data and find those 6 samples again. And that essentially allows you to do 6 samples roughly for the same cost as 1. You get less cells in that case, but you get a lot more samples per cost. So those are things that we like to discuss during this consultation step. We do FACS for you once you bring us the cells. Again, the cell dissociation part is your responsibility. Everybody has different samples, different tissues, different species. So it's your responsibility to isolate those cells and get them into a single cell suspension as possible. We have dissociation protocols that we can help you with. That we've developed over a number of years for different projects, and we can kind of help you, give you ideas on how to approach a particular tissue to get them to the best single cell suspension and also the highest viability. We do FACS to get them really to single cells. We also sort for live dead stains, so we get rid of all the dead cells. As you can see, it's very expensive to do this, and the last thing you want to do is sequence dead cells. And so we get rid of all those dead cells, and that really increases the quality of your data. We apply it to the 10x genomics instrumentation. That's a very rapid process, and that generates the libraries, just the libraries at that step. We can then return the sample to you at this point. You can sequence it yourself, you can outsource it to somebody else, or we can do it for you in the core, or hand those libraries off to the high throughput sequencing facility. And then the data processing is very similar to what I talked about for the bulk RNA sequencing, where you can do it in your lab if you have the expertise, or we can connect you with somebody in the BARC to establish a relationship for the downstream data analysis. Let me see if anybody's got any chats. Nope, good. So just as an example of this, we did this, we published this paper this year that we did a proximal distal survey of the healthy adults, human small intestine and colon epithelium by single cell transcriptomics. This was done from 3 different human donors. We got transplant organs, so the entire gut. We did 6 regions for each one of those 3 donors, so 18 different samples. And we use that hashtagging technology to enable us to do all that with 3 samples instead of 18. We could have not afforded, if you think about $8,000 times 18 versus what I just told you, is a big price difference. So this is just an example of you can do some very big studies if you design your experiment appropriately from the very beginning. How about if you can't afford RNA sequencing, or you just don't need that amount of data? We can also do high throughput qPCR. So there's kind of this wiggle room where you have, more than one gene you want to detect, but you don't want it to detect 23,000 genes. So there's a big difference in between, and that's where this kind of comes in handy. Is if you have 20 or 30 genes and you have quite a few samples, but you only want to look at 10 genes, that's all you're really interested in, this is a good alternative to RNA sequencing. I would say the biggest advantage is most people can analyze the data themselves, because what you get from this is you get CT values. You get the same value, same sort of data you get from a standard conventional qPCR instrumentation or technology. But the big difference is this is done on a microfluidic chip. You need very little RNA. So if you have precious samples, you have low input samples, things like that, you can run this on this chip because it uses nanoliter volumes here. This is just an example of the chip. And you can see here on the right-hand side a blow up of the where it says S. That's actually the test tube. I'll call it a test tube where the reactions are performed. And it's nanoliter quantities of fluids that are injected through these microfluidic lines that have the enzymes, that has the probes, that has your cDNA, things like that. This is an example of the different conformations of the chip. So what these numbers mean, this means 48 samples and 48 different genes or targets that you're interested in. It's an example where you have 192 samples and 24 different genes, 96 samples, 96 genes. And this is the number of reactions that actually are performed on the chip. So you get quite a bit of data from this technology. There are some other newer chips that I don't know if I have in this presentation, but they're a little more flexible. So you can do 12 by 12. You can do a few more different configurations depending on your needs. This is just an example, again, of the little test tubes. You can see in yellow here on the right, this is where the reactions are performed, and the blue are the microfluidic lines that automatically put the chemistry into those chambers to do the reactions. It only takes about an hour to do the actual reaction, similar to a standard or conventional qPCR. This is that chip and hole, and you can see this is at cycle 0, and then when you do the entire reaction, you get different intensities in those little squares, and the machine will read that intensity. The higher or the brighter it is, the more genes gene material you have in that particular well, and then it will give you a CT value over that entire 40 cycles. And it generates heat maps like this of the CT values, and you can download .csv files to do standard, analysis using those CT values. The workflow for this is similar to the RNA sequencing in some respects. You still isolate your RNA, but what you have to do is you have to define your own target genes, because we don't know what your experiment is. We don't design the actual biology of your experiments. We'll help you with the design of the approach, but we're not going to tell you about the biology. You have to tell us that. So you have to tell us the target genes, and you drop your samples off. If you want, you can make cDNA from this. You can give us cDNA or RNA, or we can make the cDNA for you. We'll do a QC step to make sure that everything looks good before we put it on the expensive part, because we don't want you to waste any money. We'll perform the qPCR and the biomark. Again, just this step, not including the QC and things like that. It's a very rapid process. It just takes an hour or two to do that. And then the data processing, data analysis is you. So this is something, again, that most labs have the expertise to look at CT values and do differential gene expression analysis. So here again, this is just different configurations. I didn't talk about the 12 by 12 in that other slide because it was an older slide, but these are the configurations of the chips that you can do. This is the numbers of samples. It gives you kind of an idea of the types of configurations of samples and target genes that you can detect. This is all on the web page. I know there's a lot of information here, so if you're interested in designing an experiment and need to know this, you can go to the web page and it's all there. These are some generalized prices. All the prices I'm showing you here, I just want to put this disclaimer out. These are approximate amounts because every experiment is slightly different. And it depends on if you're a UNC, NCSU, or CGIW member as well. All the pricing for non-members is on the webpage, if you want to go look at that. But this just gives you an idea about how much it costs to run this high throughput qPCR. Oh, very important. People ask this a lot. What about the probes you use? The instrumentation allows you to do cyber green. But early on, when we first got this, I made an executive decision that we were only going to use TACBAN probes. And the reason for that is because you can see it is expensive when you get up to these higher gene numbers. And if you bring us your probes and you have your cyber green probes and you haven't validated those probes and you get primer dimers, then your entire experiment could be ruined, and you're going to be out, $3,400, up to $3,400. So the TACBAN probes reduce that chance of getting primer dimers by a lot. And so, in fact, it usually doesn't happen at all for us. And so this was something that I made an executive decision for. So TACBAN probes are more expensive, as you know. What we've done to decrease the cost for the users is we call it a virtual probe bank. And essentially what this is when you come, we have a number of probes from mouse and human that you can tap into. You don't have to buy the whole tube. You can just buy a reaction. And that decreases the cost a lot. And we have hundreds of different genes from mouse and human. Now, your gene of interest may not be in there. In that case, you would buy the probe through the core, the whole tube. We would keep it in our bank. And every time somebody uses the probe you bought, we give you a credit back for future services. And so this allows you to kind of like look at our bank and say, hey, I actually have 20 genes I'm interested in. You guys have 18. And so we'll let you tap into those 18 for a really reduced price, and you buy 2, keep them in the core. And that allows the price to be reduced by a lot for doing these high throughput qPCR experiments. The next thing we're going to move on to is protein detection. I'm not going to spend a lot of time on this. It's a service that has some more conventional assays like ELISA, multiplex B technology, for instance, to detect cytokines in blood or in your supernatant from your cells, and also plate reading technology. The prices vary for this, and if you want to us to do any of these technologies, you can contact Carlton Anderson for a consultation on experimental design and pricing. Again, all of this is on the webpage. You can go to the link for immunoassays and click on that, and all of these technologies will come up. One of them I do want to spend a little more time on because it's something people aren't familiar with is single cell Western blots. The company Protein Simple developed this instrumentation. It's called the Milo. I don't know why it's called that. It's not an acronym. It's somebody's name, I think. But it's called the Milo single cell Western blot system. And I put this picture up because it shows you what the format or the form factor is for this. It's a slide. It's exactly the same size as a slide. And what it has on that slide is a polyacrylamide gel, just like you would use for a big Western blot. But it's on this slide, and each one of those little lines is, I think it's 20 or 25 individual wells in each one of these lines you can see here. And so what you essentially do is you bring your single cell suspension, or we can fact sort cells for you. We apply it, we let them settle down in those wells, wash off what doesn't get into one of the wells, and then you run it just like you do with standard Western blot. It runs through an electrical field in a denaturing solution. And then we take that slide after you've run your samples, your single cell samples out, we can probe it using antibodies, fluorescent antibodies. And here's an example we did in my lab we're interested in the role of SOX, transcription factor SOX9. And you can see here that we can detect the SOX9. This is the corresponding GFP levels in a mouse that is expressing a reporter gene for SOX9, and then beta-tubulin is internal control. You can see you can detect from a single cell these proteins. I would warn you that this is not a very routine sort of like workflow like the single cell sequencing or the bulk sequencing. This is something that is a lot more exploratory. It takes a lot more kind of working through the conditions to make sure it's going to work for your particular protein. So this is also something that if you want to tap into single cell protein detection, it's going to be a pretty extensive consultation about what your goals are, what your targets are, how highly expressed they are, things like that. And then we'll probably do a bunch of pilot experiments to make sure it's going to work for you before we engage in a larger project. Joseph.
How many targets would you expect to be able to be probed for this?
You can do up to three at a time, given our scanning technology. So basically it's the color of the secondary that you use that dictates how many we can do. So we have a scope that scans the entire chip and then compiles the entire image. And then you can go to each well and track, for instance, target protein 1, target protein 2, target protein 3, overlay those. And then the software will give you these intensity charts you can see for SOX9, the GFP, and the tubulin here to quantify the intensity of those.
Yeah, for both this and for the single cell RNA-seq, what's the minimum number of cells that you work with?
Yeah, so the chip itself has, I think, a total of 10,000 spots. And the way the cells are seeded into the individual wells is done randomly. So you basically take a certain number of cells. I think that's usually between, it depends on the cell type, things like that, but it's usually about, I think, 20 to 100,000, depending on the size, the shape, buoyancy. There's a bunch of things involved in that. So you can go from, let's say, well, actually you could do even less than that. It doesn't really matter how many you start. You could start with one, but you're not going to get much data. Right. You may not capture a cell. So we typically say, between 10 and 100,000 starting cells. We'll apply those, let them settle down. Typically you get, I don't know if I have this on there, you typically get between 1 and 2,000 cells that actually go into a well and that you can analyze in the end. So even though it has 10,000 spots, not all of them get seeded, and you don't want to load too many because then you get multimers in your wells, and that's not something you want if you're doing single cell protein detection. Yes.
Do you want to come in the same sample, or can you split it up a little bit from having multiple samples?
You can't. Yeah, they have. They have a new cassette that you can lay over this entire slide that allows you to do multiple samples. We have not used that yet, but I think you can do up to 2 samples per slide. I'm not sure on that. You'd have to look on the webpage because it is a newer technology. This cost for each chip is about $500 for members, roughly. That's not including the antibodies. Sometimes you're gonna have to buy your own antibodies, vet your own antibodies, make sure that you have a single band on a normal Western blot, things like that. So we don't want to get into too many details about that, but if you're interested, please contact us and we can go over the details of what you'll need to do for us before you bring your samples.
I'm monitoring.
Pardon?
I'm monitoring, just so you know.
Okay. The last service I want to get into is our newest service. This is a cell culture service. We call it cultures. And essentially what this is it's, most people have heard of organoids by now. And it's essentially primary tissue derived stem cells that we can culture in multiple formats. So it's not cancer, it's not transformed cell lines. These are cells that you isolate from whatever tissue you're interested in. Typically they're epithelial tissues. Most of the organoid technologies use are initiated or are started with epithelial stem cells. And they culture into 3-dimensional spheres that are hollow in the center. Those are called organoids. And they are stem cell driven. They can differentiate into the post-mitotic lineages. They can keep a stem cell compartment and a differentiated cell compartment. But also, we can grow these as 2 dimensions. So that's why we encompass all of that sort of 3-dimensional, 2-dimensional culture systems into the word culture, just to define that. Again, primary tissue-derived stem cell-driven cultures. You can get these from different species. We've grown organoids from human, mice, horses, pigs, a number of different species using this technology. And we've also cultured them on two-dimensional systems. So essentially the way this works is you have a donor organ or tissue. Again, this can be from different species. I'm focusing here on intestines, but you can do this from I don't know if I have this. No, I don't have it here. You can do it from the small intestine, the colon, pancreas. There's liver. There's a bunch of different tissues that are amenable to three-dimensional organoid cultures. In the intestine, we isolate crypts from either biopsies or whole pieces of larger tissue or organs. And of course, the crypts have the stem cell compartment at the bottom. That is essential for organoid or two-dimensional epicultures. You have to have a stem cell compartment. And then we apply these stem cells to either a three-dimensional culture system, which is a hydrogel dome, so it's three-dimensional, typically between 15 and 25 microliters. It's a droplet of extracellular matrix that solidifies or polymerizes, and it stays in that dome shape, and the stem cells are encapsulated in that. They will continue to proliferate and grow into these three-dimensional spheres given the media conditions that we supply to them. And they'll keep growing. They'll grow indefinitely as long as you feed them and you split them and things like that. We can also take crypts, we can apply them to an engineered extracellular matrix in a conventional two-dimensional format. Standard wells that you normally would use, 6 well, 24 well, 48, 96, etc. And this is more favorable for some types of experiments. As you can imagine, it's much easier to image because you don't have things in 3 dimensions to try to quantify. It's very challenging to do that. And so a lot of people like to use this 2-dimensional system a little bit better than the 3-dimensional, but there's different applications and different reasons why you might want both. And then we can also, apply these 2-dimensional culture systems to multi-well formats. So if you're doing anything in high throughput, we can put them into higher throughput well formats. Yes.
What do you mean by engineered? What do you mean by engineered?
It's what I mean by that. It's a collagen-based extracellular matrix, but we have engineered the properties, the stiffness properties of that, the polymerization properties, so that it maintains the stem cell compartment. So there's a number of papers we've published with a collaborator, Nancy Albrecht, to show the different stiffnesses of these extracellular matrices will either support or not support stem cells. So if you, as I mentioned, you have to have a stem cell compartment for these cultures to work. And if you have too stiff of a matrix, and that's what I mean by engineered, it's engineered to be the right stiffness that it can maintain a stem cell compartment. Now, what we can do with these 2-dimensional or 3-dimensional systems is they're primarily expanded as stem cells because those are only cells that are dividing. So we keep these cultures initially in medias that will promote stem cell division or maintenance. And that expands the amount of cells you have. You can do that in 3D and 2D. But if you're not interested in studying different or undifferentiated or stem cells, you're interested in the differentiated compartment, we can then switch the media to a media that will promote the stem cells to change into their post-mitotic lineages. And we have different media formulations that will do that for intestine and colon. If you're looking at something that's outside of those 2 tissues, that's something that will be a little more exploratory because that's not something we do routinely, but it's something that we can accommodate.
You guys played with like intestinal co-cultures, like adding other cell types beyond just that epithelium?
We haven't. That's something that we could certainly do. It would be outside of our normal kind of operating procedures, but it's something we could certainly engage. The idea here is this is a challenging technology because you have to have all of the right things, and there's a lot of things that go into the media and go into the extracellular matrices that have to be absolutely perfect in order to get reproducibility. And so that's something that if you're just doing this as a one-off experiment, it's not really feasible for a lab to get up to speed with that technology to do it efficiently. It's going to cost a lot of money. There's going to be a lot of failure. So that's why we offer this service, because we do it all the time. And the idea is to give you those cells and then let you do the perturbations. We're not doing the perturbations for you. We're just getting the cells to the state that you want them, and then you do the perturbations. So again, this will go, if you were interested in this, the cell culture technology, apiculture technology, something you have a consultation for, we'll discuss your experimental design, outcomes, goals, things like that. And that'll help us kind of inform you on how to approach what you do. Do you do 3D? Do you do 2D? Do you do multi-well? Do you do little wells, big wells? What kind of differentiation media might you need? Do we need to do pilot experiments to make sure you're getting the post-mitotic lineage that you're expecting? All of those sorts of things. Did that answer your question?
Yes.
How much time it takes to develop the 2D monolayer in the
This one right here,
yeah.
you need to confluence.
Yeah.
So it depends. It depends on how big the well is and how much you initially seed. But if we do a standard passage from a confluent well to a split or 1 to 3 split, let's say, it'll take a couple days, maybe 3 days to get to almost confluence again to just split them. Depends on the cell, depends on the the tissue, the organism. There's a lot of things like that that change the growth dynamics of those stem cells, but doesn't take too long typically. Did that answer your question? You have a question? So that's this ep culture service. Now we have the core has a tissue bank. So a lot of people don't have tissue to work with, but they want to use these cells because they're very powerful. They're not cancer cells. And so if you're interested in normal cell biology and you want to use these, we have some tissue that you can use. And where we get this is from intestine and colon from humans right now. And where this comes from is we get organ transplant donor tissue from it's called Honor Bridge. It was otherwise known as Carolina Donor Services. And since the intestine is really not transplanted, we get that as a research organ. It's very fresh, and we isolate stem cells from 6 regions along the axis, proximal distal axis, duodenum, jejunum, ileum, ascending transverse, and descending colon. And we have indefinite supply of stem cells essentially in liquid nitrogen that you can tap into. Here's an example. I think we've got, here's 2 that we don't have any information for, but we've got about 14 different cases right now of different demographics. So a mix of male and female, different ages, and different races and ethnicities that you can tap into. So if you're interested in biological replicates for a human study, and you want to tap into 3 different people and then perturb something, we can grow different donor cells on this platform. So this is something you can ask us to give you these demographics, and you can select and choose not only different donors, but also different regions to apply. And again, I told you before that we've done this with multiple different organisms: mouse, pig, horse. We've even done it with zebrafish, at least a 3-dimensional organoid culture. So the culture conditions are amenable across different species, is what I'm trying to get at here. So as I mentioned, this is a new technology, and we're phasing in different parts of this technology just because it was too much to do at once. And what we have now, we've completed phase 1, and this is the conditioned medias. These are the medias that you apply to those stem cells to either keep them as stem cells and expand them in the culture or differentiate them. And these medias are not, they contain factors that promote stem cell maintenance. So for instance, things that drive Wnt signaling. And it's very expensive to use recombinant factors. So we have conditioned medias that cell lines make that increase those particular basically three factors. And we have some small molecules we put in the media that will promote stem cell maintenance. And we provide this media for users. If you want to do this in your own lab, we can sell you the media, or we can do it in our core facility. So this is available currently. Then we've most recently introduced these particular services: organoid culture, self-renewing monolayers, differentiated monolayers, ep culture biobanking. What I mean by that is if you have a tissue sample that you would like us to expand as stem cells for future use, we can take that biopsy, we can take a mouse intestine or colon, and we can generate 3D organoids or 2D monolayers and then freeze them down for you, or expand them for your experimental downstream applications. And then ep culture training. So this is a big thing. We don't have a lot of hands in the core if we have too much demand. One of the things we do want to do is we want to kind of export or get this technology out into the field so people can do this in their own labs. And so we do offer training services for these ep culture cultures. And then the last thing which we have not implemented yet is gene editing. So a lot of people want to say, hey, I'm interested if I knock out this gene, what happens to my cell type of interest, or if I overexpress a gene. And so in phase 3, what we're going to do is we're going to offer gene editing services. This is the plan. I'm going to show you a slide in a minute what we've done to kind of get this rolling. Pricing for this varies. This is a very new technology. It's a very new service, and we haven't worked it. Again, every project is slightly different. It's going to take a different time, different cost structure for each project. So this is something in the consultation step we will go through in detail and then give you a quote for how much it will cost to do a particular project. So again, this gene editing, we published a paper this year that describes efficient transgenesis and gene editing using CRISPR technology for human intestinal stem cells from the small intestine and the colon. So we've got a very nice way of either making random integrants using back transposase, so piggyback transposase, or CRISPR-Cas9 gene editing. And we can do this very efficiently on the 2D system. And that's, again, a little bit more amenable to just normal procedures that you would do in your lab. It's very fast and very efficient. And here's just an example where we took a colony of human jejunum stem cells, and we introduce a constitutive GFP that has basically an NF-kappa B luciferase responsive element in it. So we can look at the response of inflammatory stimuli through NF-kappa B in these human stem cells. Or we can differentiate those cells into, for instance, absorptive enterocytes or goblet cells or some other secretory lineage, and we can assess how the inflammatory stimuli impact those particular lineages. So I'm going to stop there. If you want to do a project, contact Carlton Anderson. He's Associate Director. His information is on the webpage. Remember, just Google CGFD AA Core. There's also a core use request form you can fill out there if you want to contact us through that particular modality. And if you do use the core, remember to acknowledge the CGFD P30 grant. This technology and the core staff are reliance upon that particular grant to offer these services. So we wouldn't be able to offer these without that. And so we need everybody who uses these to remember to acknowledge that in their grant applications and also their publications. And Amy wants to say something.
Yes, I do. And you can find the actual grant number that you need to cite on the CGIBD website.
Yes. And I'm going to put it in my signature line probably, so that every time you be in Carlton
yes.
for staff. So if you forget, you can just find their email
yes.
and then go look for that grant number to cite. I should have put it up here, but nobody would have written it down anyway. So.
I know, but I just have to say that.
Okay. With that, I can try to answer any questions that you might have.
Okay, nothing on the chat. Chat is clear. Anybody who is remote, feel free to just unmute if you have a question as well.
On the bulk qPCR chips, is it any cheaper if you don't fill the chip, or is the price by the chip?
The price is by the chip. Once the chip is run, you can't reuse it. Okay. So yeah, so you want to try to fill it up, and that's something, you know, if you have friends or you have, you know, other investigators that want to do something, you might want to like collaborate with them and say, hey, I have 6 samples, you have 20, you have another 20, and then you can fill up the chip that way as well.
What's the, so you mentioned with the single cell Western stuff, like there needs to be pre-validation, right, with the antibody testing and everything. What's been the translatability of that so far? Like, does it need to be like really robust? Is it, you know, like what's the confidence in translating from?
That's a hard question. So the question is, for the single cell Western blot, one of the pilot experiments you need to do in your lab is to validate your own antibodies. And because you don't get good resolution between proteins on the single cell, you get basically a blob. You can't distinguish if you have multiple bands if they're there. So you have to make sure in a conventional Western blot that you have a single band. That's the best way to validate your antibody. It's hard to compare those two because we can't ever go to the single cell and resolve small differences. If you have differences like that are like, 20 or 30 kilodalton between your proteins, then you can usually see that on this technology. But if it's less than that, if you have 3 bands and you're already picking the one that you know is the right size, you're probably not gonna be able to do that if they're really close.
So probably also not like super level for food proteins with like numerous conformations and.
Exactly, yeah. So that's one thing I will say about the single cell Western blotting is that the resolution of detection between bands is not really precise. And so you really have to know that you have a nice single band. If you're going to detect multiple proteins in that cell, that same cell, they have to be separated by quite a bit. Now, I will say that if your proteins are very close, and you're interested in, like, say, 2 proteins that are really close, you can probe for one, you can strip the chip and reprobe for that second one. So that's something you can do if your proteins are really close to each other. And I think you can strip it up to 2 to 3 times, they say, without losing a lot of sensitivity. Again, that depends on the protein and things like that. We haven't run a lot of these. I've run probably the most for my own research program. And it depends. It's extremely variable how well things work. We've had things work in conventional Western blots or immunostaining really well, and it doesn't work on here, and vice versa. So I think you really have to just decide if it's an application that you really need, and then we can work through the details with your antibodies. Anything else? All right. Well, thank you.
Thank you, everybody, for coming. Thank you all that came to in person.